Hi guys, my name is Vaishnavi. In this video, I'll be talking about structure of the tongue, an overview of the salivary glands of the oral cavity. The tongue is a mass of striated muscle fibers covered by a mucous membrane whose structure varies according to the region. The tongue's dorsal surface is irregular and covered anteriorly by a great number of eminences called papillae. The posterior third is separated from the anterior two thirds by a V shaped groove called the terminal sulcus. Behind this boundary is the root of the tongue, whose surface shows many bulges of the lingual tonsils and the lymphoid nodules. There are four types of mucous membrane elevations, which are covered by stratified squamous epithelium. The first one is filiform papillae. It's very numerous, heavily keratinized. Their epithelium lacks taste buds. Their rough surface facilitates food movement during chewing. Fungiform papillae. It's present among filiform papillae. They are less numerous, lightly keratinized, mushroom shaped, and have scattered taste buds on their upper surfaces. Foliate papillae. They are poorly developed in adults. Valate or circumvallate papillae are least numerous and largest lingual papillae with taste buds. 7 to 12 circumvallate papillae normally form a V shaped line before the terminal sulcus. Ducts from several serous salivary glands empty into the groove that surround each papillae and wash food particles from the vicinity. Taste buds are ovoid structures. About half the cells are gustatory taste cells. Other cells are supportive cells, immature cells, and stem cells. The base of each taste bud rests on a basal lamina and is entered by a afferent sensory axon that forms synapses on the gustatory cells. At the apical end of the gustatory cells, microvilli project through an opening called the taste pore, the place where molecules dissolved in saliva interact with the cell surface taste receptors. Salivary glands. They are exocrine glands, and the main function of salivary glands are to wet and lubricate ingested food, initiate the digestion of carbohydrates and lipids with amylase and lipase, secrete protective bacteriostatic substances such as IgA and lysozyme. There are three pairs of large salivary glands: parotid, submandibular, and sublingual. There are two major kinds of secretory cell: serous and mucous cell. Serous cells are protein secreting cells, usually pyramidal in shape, with a broad base resting on the basal lamina and a narrow apical surface facing the lumen. Adjacent cells are joined together and form a spherical mass of cells, which is called acinus, with small lumen in the center. Serous acinar cells largely produce digestive enzymes and other proteins. Mucous cells are more cuboidal or columnar in shape, with nuclei pressed towards the base of the cell. They have mucus secreting cells, which contain hydrophilic glycoprotein mucins, which are important for the moistening and lubricating function of the saliva. The mucus cells are mostly organized as tubules rather than acini and produce mucins. Exocrine glands have a secretory portion and a duct portion. Ducts can be simple or compound, and the secretory portion can be tubular or alveolar or acinar. In the intralobular duct system, the secretory portions empty into the intercalated ducts, which are lined by cuboidal epithelial cells, and several of these ducts join to form striated ducts, which have radial striations. The striated ducts of each lobule converge and drain into the ducts located in the connective tissue septa, separating lobules, where they become interlobular ducts. In the large salivary glands, the connective tissue contains many lymphocytes and plasma cells, which release IgA that form a complex with the secretory components synthesized by the epithelial cells of serous acini and intralobular ducts. The IgA secretory complex released into the saliva constitutes an immunologic defense mechanism against pathogens in the oral cavity. Parasympathetic stimulation through smell or taste of food also produces secretion of saliva. Parotid gland. It's located in each cheek near the ear. It's a branched acinar gland with secretory portions composed of exclusively serous cells. The serous cells contain secretory granules with abundant alpha amylase and proline rich proteins, which have antimicrobial properties and calcium binding properties that may help maintain the surface of teeth enamel. Submandibular gland is a branched tubuloacinar gland with secretory portions containing both mucus and serous cells, but mainly serous cells. Most of the secretory units in this gland are serous acinar, with some mucous tubules capped with serous cell. Such capped cells are called serous demilons. In addition to alpha and proline rich protein secretion, the serous cells of the submandibular gland secrete lysozyme, which hydrolyzes the wall of many types of bacteria. Sublingual gland is a branched tubuloacinar mixed gland formed of serous and predominantly mucus cells. It's the major salivary product is mucus, but it also secretes amylase and lysozyme. Thank you.